My name is Stefanie Kiderlen, and today I will present to you our company, Prospective Instruments, as we are one of the newest members of this industry board. And yeah, I will first give you a short overview about our company and of course some examples, some user examples of my microscope. I will introduce to you one of our microscope series, the MPX series, and at the end tell you something about our in-house imaging service. And I will start with a quick overview about our company. So we are a startup located in Dornbirn, Austria. And our CEO, Dr. Lukas Kreiner, founded this company in 2019. And as you just mentioned, we focus on turnkey, compact, easy to use, multimodal microscope solutions, which I will tell you, where we'll tell you something about later in more detail. And with our microscopes, we are trying to be vertically integrated with respect to the light engines, mechanical design and system control, which means our microscope completely integrated, which we'll see later. And we started our in-house imaging service where we can offer more imaging techniques than are in the MPX series right now. But I will give you an overview about the in-house imaging at the end of this presentation. Here's an overview about our core competence. Because of time issues, I won't go into much detail for every point, just that you have an impression about our core competences. So as I just mentioned, we developing microscopes, of course, but we are also developing light sources, which means hopefully 2022 you can also buy femtosecond lasers as a standalone devices. They're not launched yet, but hopefully soon. And we're providing OEM solutions for multiphoton microscopy. Of course, we have a high resolution microscopy techniques. One big point is, of course, label-free imaging. Most biologists don't like this this word autofluorescence, not so much, but in this case, it's an important thing because we are also doing deep learning image analysis, which means that we use this autofluorescence to create H&E, virtual H&E stainings with our medical device series. I won't review about this series today, but maybe in another virtual pub. So today I want to introduce to you our MPX series. We have 3 standard versions, which means that the customer can decide between 3 different wavelengths, 1 fixed wavelength, 2 fixed wavelengths, or tunable wavelengths for the multi-photon excitation. And of course, you can choose or upgrade your microscope with a lot of options and accessories, which maximizes the utility of these microscopes and tailors the specific needs and one important thing, of course, the budget of the customers. This is our MPX microscope. This is the first version of our microscope. We already had a design revision, but the principle of this microscope is still the same. So our microscope consists of only 2 components. You can see the scannet over here, which is connected by a flexible umbilical to the controller over here, which gives you a high flexibility for this microscope. The scannet is mounted onto an XYZ stage, and which gives you a large working distance. And you can choose for the scan head upright or an inverted configuration with just a very simple modification. And also in the future, you can we plan to integrate a horizontal tilting of the whole scan head. So our microscope is designed to address multiple experiment requirements, which means that the person uses this microscope can just modify the microscope and don't have to modify its the experimental setup, actually. As I mentioned before, the microscope is fully integrated, which means you only have these two parts. You don't have external light sources or an external PC standing around. So everything is really integrated into these two components, the scanner and the controller. And this makes the whole microscope very flexible. I will give you later a few examples of our users of this microscope, how simple it actually is to build the microscope up, and how it works just out of the box. And yeah, the microscope is also multimodal, which means we have different microscopy techniques combined in the microscope. Right now we have multi-photon microscopy, the higher harmonics generation microscopy, single photon microscopy, and in 2022 we will also provide fluorescence lifetime microscopy in one device. So this multimodal imaging also gives you a multifunctional application, actually. So here's an overview about different applications. You can image with just one microscope. You can do, for example, whole slide imaging or live animal imaging, but you also you can do the classical immunostainings and, as I mentioned before, label-free imaging. And of course, 3D imaging, which means also deep tissue imaging, which is important, for example, for the mouse imaging. You can't clear a living mouse, right? But you can do this deep tissue imaging with cleared and non-cleared samples. So you really have a high flexibility regarding from the size what you want to image. You can do the whole slide imaging, for example, 2 × 2 centimeters or even larger. You can image just one single frame, or you can go to even the single cell level. And you also have the high flexibility by choosing your sample. As I mentioned before, you can, of course, image the gold standard, which are FFPE samples, because they are very thin and good to handle. But you can increase the thickness of your sample by using, for example, cryosections or even 3D tissue. And you can choose between a label-free imaging, which gives you, in some cases, much more information than probably you would think. For example, the high harmonics imaging is in the multi-photon modality. It's suitable to image collagen fibers very nicely. And but you can also do the classical immunostainings or other stainings, and use different samples, different thickness, different sizes of your samples with the microscope. And you also have the high flexibility regarding the working distance. Here I show you some different setups that we use with the microscope. Because you move the scanner and not the sample, or actually you can move both if you want. You have the high flexibility to, for example, do a live animal imaging, to put a living mouse under the microscope. You can do 3D imaging using some 3D tissue. In this case, there were some mouse organs. Or you can do the classical imaging using microscope slides, of course. So you have you can choose your working distance. And yeah, which brings me to some customer experience examples. Sorry. In this case, our microscope was in the lab of Bruno Weber in Zurich, where they do the live animal imaging. And as you can, on the left image, you can build up the microscope very easy, as you just have these two components. And the stage under the microscope where the mouse is fixed on, this is the stage of the lab they usually use. So you can design your experiment as you want, and you just can modify very easily the microscope, and you don't have to do it the other way around. And yeah, these are some results of the live animal imaging. As you can see here, it's a mouse cortex, a time lapse of a mouse cortex, where you can see the neurons in green and the astrocytes in red. And the neurons are labeled with a calcium indicator. So every time when the neurons are blinking up, it means that there's a calcium influx of the neurons. And of course, you need a kind of special robustness. I can say like this for the microscope. And this is a very nice example for the easiness how to use the microscope, because during the COVID lockdown, one of our users, Professor Rami, he took the microscope just to his home office. So he packed the microscope in his car and built it up in his living room, actually. And he did the imaging at home. And he did some quite good quality of the imaging. As you can see here, he was imaged some pollen grains and also mouse cortex, and just in his home office, actually. And here's an example for our inverted setup. As I mentioned before, you just can invert the whole scan up at the whole scan head. And this is an example of Professor Heiden's lab in the Translatum in Munich, where they have a microfluidic system, which you can see on the right side over here. And as you can see, there is a big pump and a lot of tubes around the whole system. Here is the microfluidics. And so it was very important for them that they have a lot of working distance. They can build up their experiment on top of the microscope. The stage is 3D printed, their own design, and they could easily build it up on our microscope. And what they want to do is they want to create, or they created with this microfluidics, in vitro system that mimics in vivo conditions, which you can see on the left side over here, as a test system for example, osmotic pressure on blood cells. And first thing, what they want to see was that they want to show their cavity they created in this microfluidics, which you can see over here. And therefore they used our multi-photon modality and did a set stack, and of the set stack you can build up this 3D projection. And as a proof of concept, they used fluorescent beads or lung cells, and in the last step even blood cells, to show that they could capture these cells in this cavity. And also they could do a nice time lapse where you can see when the cavity is built up by piezos, when they start the piezos, you can see the beads captured in this cavity, and then they turn it off, the beads are gone. And yeah, this was a very nice example for the inverted configuration of the microscope. But we have another user which was very interested in this upright configuration, especially for the water dipping objective with a long working distance in the Kanter lab in Munich by Dr. Stefanie Kiderlen. They want to image tissue engineered constructs, in this case an artificial artery, which you can see here on the left side. So this is a normal Petri dish, and you can see the artery here in the middle. This is a scaffold which is covered by GFP labeled cells, and they want to image this artery. But because of this artery is hanging up in the medium, it's a little bit difficult with normal objectives, so you need a long distance objective, and they use the microscope to do this set projection of the artery, and which worked actually very nice. But you can see that, of course, you're on a special point, you are limited in the depth of where you can image. And this was up to almost, I think, 1 millimeter. But yeah, and then you only can see one side of the artery. What we did then was we tried to image the artery in the mesospin light sheet, and which you can see in the 3D projection. I must say it's not the nicest image actually, because you can see we had some issues with the preparation of this artery. We have to work on this, but just to show you that, yeah, even if you need larger images, you just can, of course, in this case, it's another microscope, it's not the same setup, but so we were able to image the entire artery in this case. And yeah, which brings me to our in-house imaging service. So we have, of course, different techniques in our in-house imaging service, like the single photon fluorescence, where we have an upright and inverted configuration, and of course, a multi-photon fluorescence. And with this, of course, 2-photon, 3-photon, single harmonic and third harmonic imaging. We will have a film setup here, which means we have the TD and the FD film setup, and the whole slide imaging setup, where you can scan very large sections. Yeah, one of our newest microscope is the light sheet mesospin setup, which is part of the mesospin project, where you can look at mesospin.org. It's a fully automated light sheet. And of course, we have additional equipment in our in-house lab. So if you're interested in some imaging, you just can contact me and we can talk about your experiments and which modality or technique is suitable for you. So yeah, thank you very much. And if you're interested, you can just visit our homepage or contact me. Thank you.